Hello, today we will be talking about the ecology and the geological distribution of corals. First, we will be discussing the ecology and then we will go into the geological distribution. Corals constitute a variety of the primitive, predominantly marine, and these animals have uh, two germ layers which is usually called as diploblastic. So, these are essentially diploblastic metazoans and are by far most significant in the entire fossil record. Despite the poor preservation of many of the coelentrates or corals, they nevertheless exist as scattered fossils which extend back probably to the late Precambrian also. Let us talk about the paleoecology of the corals. Modern corals range in space from the tropics to the higher latitudes, higher means more than 75 degrees, and from the surface to the abyssal depth. That means we have defined their distribution in space in two dimensions, one in the horizontal plane, that is up to the higher latitudes greater than 75 degrees of the latitude, and then in the depth. So, this is in the vertical plane. So, vertical plane it has or it witnesses the presence of the corals from the surface to the abyssal depth. Two ecological groups are recognized, one with zooxanthale or with the symbiotic algae and the other without any symbiotic association. Corals with aforementioned symbiotic algal association dominate present day coral reefs and are confined in their distribution by their algal symbionts to the water temperatures of about 16 to 40 degrees Celsius and shallow water depths, usually less than 190 meters. Other corals often exist through a much broader range of the temperature and depth. They are minor components of shallow water tropical reefs, but build structures of their own thickets and blanks in deeper and cooler waters. The primary functions of the symbiotic algae zooxanthale is not very clear, but they form a significant fraction of the energy budget of the coral, because the algae is photosynthetic in nature, that contributes to about 98% of its fixed carbon requirement. They appear to enhance calcification. However, many massive corals appear to have reduced growth rates, and recent evidence suggests that in this case zooxanthale may act to switch the calcification on and off rather than accelerate it, thus conserving energy. The algae also remove waste from the coral and may thereby allow very large colonies, large means more than 10 meters, of tiny individuals which are approximately 1 millimeter in diameter to develop. There is a marked contrast in coloniality between zooxanthalate and azooxanthalate corals. The symbiosis within algae appears to have developed most probably in the early Jurassic. A major factor in the success of the corals in building rigid structures is their ability to cement the skeletons securely to other skeletal materials and to encrust hard substrates. Shallow water Scleractinian dominated reefs became important from the Jurassic onwards, although deep water blanks built by azooxanthalate Scleractinians extend back into the Triassic. Rugose and tabulate corals could afford neither the advantages of the algal associations nor the ability to encrust securely to the extent seen in Scleractinians. However, Scleractinian zooxanthellate colonial corals show flattening of the skeleton and significant reduction in the growth rate with depth as light levels decrease. A recent study of a widely distributed tabulate coral among those considered most likely to have harbored algal symbionts showed that no signs of these effects. In addition, only 35% of rugosans, but all tabulates are colonial, and massive growth forms of any Paleozoic coral appear not to exceed about 2 meters in diameter. Apart from some specialized forms, rugose and tabulate corals showing secure encrusting attachment or overgrowth as mature coralla are rare. This reflects the presence of an external wall or epitheca or holotheca around the corallum and the lack of an extensive edge zone of the soft tissue, the reverse of the situation in most Scleractinians. Instead, the Paleozoic corals were predominantly adapted to the life on softer substrate. They were subsidiary contributors to reef cores, although common in perireefal communities, and reached their greatest importance in bioherms and biostromes, with little evidence of frame building, and were often associated with mud-grade carbonate environments. Many show evidence of instability and smothering as soft substrates were disturbed by storm activity and sediment movement. They reached their maximum diversity in warm, shallow waters, with diversity decreasing with depth and towards the contemporary poles. In addition, there was distinctive low diversity community of small, thick-walled, tabulate and solitary rugose corals, characteristics of dysaerobic, dysphotic and other marginal environments that persisted from Silurian to the late Permian. The lacophyllid fauna, I mean to say. It was characteristically cosmopolitan with comparison to the provincialism of the other coral faunas. So, we have discussed the ecology, and in this ecology, one of the most interesting feature I would like to share with you is that you know that the earth revolves around sun in 365 days, which is what we call as 1 year. But do you know, in Devonian there were 400 years in a year. This time we have 365 days in 1 year, but in Devonian we had 400 days in 1 year. Well, how do we know that? Actually, the coral Calceola, it secretes one calcareous plate every day. That means a coral that is 1 year old now will have 365 plates, but the coral in Devonian have 400 plates. That means the year in that time had 400 days. Now, let us discuss geological distribution of the corals. This is the next topic which we are going to discuss. Fossil corals are comparatively rare in argillaceous and arenaceous beds, but often abundant in calcareous rocks. Many limestones being formed largely on the basis of the coral remains only. This is indeed what might be expected, since existing forms can, as a general rule, live only in the clean water. All three of the principal classes have been claimed to be present in the late Precambrian or Precambrian Ediacaran fauna. Precambrian time is late Precambrian time is also called as Vendian or Ediacaran. Here, internal and external molds of non-mineralized organisms are preserved in siltstones and fine sandstones. They are controversial, and alternative interpretations have been advanced for these fossils. However, some are convincingly cnidarians as well as unassigned jellyfishes. There are other or likely of the affinity Scyphozoan affinity, for example, Kimberella, and structures reasonably interpreted as the floats of hydrozoan conifers or Ovatoscutum and Chondroplon. The most striking anthozoan is the sea pen or the Carneodiscus. Other structures might represent the remains of cnidarians that are now extinct. Unfortunately, even assuming their correct interpretation, these remains to throw no light on the relationships within the cnidaria, but it suggests an earlier period of diversification not represented in the fossil record, at least to date. The origin of the classes has to be inferred by indirect means and has been the subject of much discussion. The internal structure of the gut and the character of the nematocysts suggest that the hydrozoa are the most primitive cnidarians and the anthozoa are the most advanced. The most favored scenario envisages a hydrozoan medusa derived from a planuloid ancestor as the ancestral cnidarian, with polyp evolving later as a prolonged juvenile stage. Now, we will discuss the Phanerozoic diversity. The type of preservation represents the Ediacaran fauna is not known after the Precambrian. This is attributed to the rise of active macrophagous predators and scavengers, disruption by burrowing organism, and possibly the increasing influence of the other fauna, all contributing to the early breakdown of soft tissue in comparable environments. The fossil record of soft-bodied cnidarians becomes sparse and patchy. Hydrozoa are represented by very few, sometimes doubtfully assigned medusa, such as questionable Circummeducina of the late Carboniferous, and the more convincing Hydrocraspedota, that is mid to upper Jurassic, and Kirklandia, that is lower Cretaceous. Hydroid polyp colonies are better represented. A rich fauna is known from the Ordovician and Silurian of Scandinavia, some so well preserved that the scleroprotein periderm can be recovered from the calcareous matrix by acid digestion. In young rocks, hydroid colonies commensal with serpulid worms have been preserved as molds and cast by the overgrowth of the basal stoloniferous network by the calcareous worm tube or Proteophylla gastroi. Dravotella, a freshwater hydroid, rare even among living hydroids, is recorded from the upper Carboniferous Mason Creek Lagestra of the Illinois. Structures interpreted as the floats of the chondrophores are relatively common in the early and mid Paleozoic. Examples are Senella, that is lower Paleozoic, previously referred to the mollusca, and Plactodiscus of the lower Devonian. Preservation is helped by the stiff scleroprotein cover surrounding the float. After the Carboniferous, however, for reasons unknown, only a single further example is recorded. The siphonophores appear to have left no fossil record at all. Finally, there are few calcified hydrozoan polyp colonies in the fossil record, which excludes the Stromatoporata and Chaetidia, both formerly assigned to the Nadeia but now refer to the Porifera. The Milleporina or Millepora is found on living reefs, particularly in the Caribbean, and Stylastrana can be traced back to the late Cretaceous. The hydroid Hydractinia, which is now most commonly found in encrusting gastropod shells, extends back to the Eocene and possibly the Mesozoic. Let us talk about the corals from Cambrian onwards now. The Cambrian form, that is Archaeocyathus, found in the Cambrian and North America, Sardinia, Spain and Australia, has sometimes been regarded as a coral, but is probably a sponge. Then coming to the Ordovician. In North America, corals, especially Streptoplasma and Columnaria, are common in this system, but in England only a few forms have been found, the most important being Favositis, you may be knowing, Heliolitis, Halicitis. Silurian corals are very abundant, especially in the Wenlock limestone. The Dugos constitute the xyloids, Acervularia, Omphima, Cystiphyllum, Rhodonophyllum, Conophyllum, Triplasma, Calostylus, Paleocyclus, Petraria, Goniophyllum. Coming to the tabulate corals, they are Syringopora, Favositis, Heliolitis, Plasmapora, Propora, Halicitis. Then comes Devonian. The Devonian corals, the rugoses include the Cyathophyllum, Heliophyllum, Endophyllum, Spongophyllum, Mesophylloides, Zephrentis, Primatophyllum, Mesophyllum. Then Calceola, to which I was talking about, that because of this is an index fossil of the mid-Devonian, and because of this fossil we know that the days had 400 days in an year. Then coming to the Carboniferous. Carboniferous rugose corals constitute the Paleosmilia, Lithostratian, Orionestraceae, Zephrentis, Caninia, and Plexus. The tabulates include Michelinia, Syringopora. The Permian corals constitute the rugose and tabulate as well, and they generally are similar to those of the Carboniferous, but have been found in Russia, China and Timor, etc. They constitute Vagonophyllum, Corvania, Zephrentis, Cania. Then comes Triassic. After Permian Triassic mass extinctions, the corals decline in their diversity. Corals are totally absent in England, but occur in the Alpine Triassic. The Paleozoic forms have become extinct, and in place of them you have now the Rhabdophilia, Montlivaltia, Thecosmilia, Stylophyllum, Stylophilopsis, Stylina, Omphalophila. Then comes Jurassic corals. The Jurassic corals includes the Stylophilopsis, Heterotraceae, Astrocecinia, and Thecosmilia are found in the lias, but are not common. In the Oolite, corals becomes very abundant. For example, Montlivaltia, Thecosmilia, Stylina, Cyathophora, Cladophilia, Calamophilia, Comatoceres. In Cretaceous, the corals are not abundant in England. The chief forms are Parasmilia, Trococyanthus, Holocystis. In some parts of the Europe, especially in the Gossau beds of chalk age of the Austrian Alps, corals are very numerous and include the Astrocenia, Montlivaltia, Cyclolitis, Synastria, etc. Then in the Cenozoic, corals are rare in English Cenozoic formations. The main corals constitute the Turbinolia, Dendrophilia, Oculina, Uniopora. They occur in the Eocene. Madrepora in the Oligocene, Flabellum in the Pliocene. In the middle and south of Europe and in the southeastern part of the United States, corals are found abundantly in various Cenozoic deposits. Thus, you have seen that we have discussed in the first part of the lecture about the ecology of the corals, and in the later part of the lecture about the geological distribution of the corals in time and space. We will be discussing about the morphology and the geological distribution of the other groups of the fossils in the lectures to come. Till then, thank you very much.